I think, yeah, good, great. So hello and welcome to my talk today. I feel very much privileged to kickstart a wonderful scientific journey for all the fellow SCIOMICS Club members and other students also. So in this short talk, I will actually break down the title word by word to finally reach the holy grail of how to build an interpretable machine learning model for precision oncology. When Subrajit contacted me first to give a talk, he gave me, I should cover three portions. I should cover how bioinformatics has changed biological sciences, and I should cover some of my research work and also some future prospects in bioinformatics for fellow students. So I would take this stage to mention that if it's not for bioinformatics, you and I would not have been in front of this computer talking about bioinformatics. One doesn't have to look far ahead or far anywhere else to notice the impact of bioinformatics and biological sciences other than the Cancer Genome Atlas data. So if you're not familiar with that, then go ahead to TCGA Atlas, and then you will know a lot more about that. And I would talk about my work in Jawaharlal Nehru University. As I mentioned, I would break this down word by word to finally reach you to a level where how we can build a machine learning model for precision oncology with interpretation possible. And with that, I would like to start by asking you all to wonder, what is science? So apart from a lot of definitions one can come up for science, a short description from another seminar I was attending as a student resonates with me, and I want to share that with you all. Science is a club where everyone is passionately discussing about something. Some agree and some disagree with each other. No one knows who started the discussion or when was it started. You enter the club, you are overwhelmed and tongue-tied at first. You stand aside and listen to the conversations. After a while, you get a grasp of what is being discussed. You formulate an opinion for yourself. Now, you feel the need to contribute to the discussion and you enter the table. You present your views. At first, no one seemed to take care. The more and more you talk about your views, people take notice. Many agree and few disagree. And over time, finally, when you feel your time is up, you leave the club while the discussion goes on and on and on. So this, yeah, I like this approach of how science is being practiced currently as such. So let's dive straight into the topic at hand. Oncology. Oncology is the study of cancer, and cancer doesn't need any introduction at all. It is the leading cause of deaths worldwide, and the death rate is predicted to increase despite our efforts in treating cancer. And our efforts in treating cancer have relied on drugs that are marginally more toxic to cancer cells than normal cells. The problem arises in such situation because on an average, only 25% of the cancer patients respond to cancer drugs. In comparison, about 80% of the patients respond to popular painkillers that are available in the market. One of the prominent solutions to this problem is precision oncological therapy, where a treatment strategy is designed by targeting molecular characteristics of the patient's tumor cells. Hence, the treatment is personalized to the patients who are likely to respond to specific cancer therapies. So, gone are the days where one size fits all, but now we accurately predict or accurately try to find out which medicine or which therapy works at what level to different patients, thereby personalizing the medicine to the particular patients. So precision medicine efforts typically involves first molecularly characterizing tumors using next-gen sequencing techniques, and then systematically studying the tumor's response to multitude of therapies in a suitable model systems. For example, depicted here are two kinds of model systems. One are cell line, in here cell lines to which multiple drugs are administered and the cell line's tumor growth is being studied. And the next is called patient-derived xenografts, which I will talk about more. So by doing this, one can identify a list of therapeutic options that is personalized for the patient. The main hurdles for this kind of therapy are, even in developed countries like USA and Europe, most of the countries, large-scale implementation of such precision medicine therapeutical approaches are not yet very well established. The time required for developing a good quality model system is about 3 years. And in such a time, 3 years, the tumor's progression could alter its response, alter its own morphological and phenotypical behaviors, and hence its response to the choice of the therapy. And also, not to say the least, privacy issues surface once we start collecting such sensitive data that could identify the patients. But the model system-based strategy to molecularly characterize the tumors and to study their responses under multitude of treatment options can be leveraged using bioinformatics to study the biology behind the tumor's response to different treatments. So how do we do that? So to understand an event or a biological process, scientists often resort to constructing scientific models. A scientific model could be a physical or a mathematical or a conceptual representation, or all the three, of the processes involved. These models help us to identify and to understand the underlying patterns by analyzing complex data. The primary purpose of such scientific model is to make meaningful predictions. So with the advent of big data and the complexity of the data increases, and also the underlying pattern becomes concealed within multitude of different patterns that are not of our interest in that complex data. So in such situations, the ability of the machine learning algorithms to learn and to improve without being explicitly programmed is being employed. And how do we build a machine learning model for precision oncology? So far we have covered what is precision oncology and why do we need models to study scientific questions. And now we go straight to the heart. So how do we build a machine learning model for precision oncology? So to any scientific model, the primary component is good data. So remember a couple of slides ago, I spoke about model systems used to characterize tumors. So we can make use of that data that is made publicly available from one such study that is published in Nature Medicine in 2015. So the tumor cells, so what happened in this study and also what happens in this model system is the tumor cells from the cancer patients are extracted and subjected to next-gen sequencing techniques, and the information about gene expression, gene mutation, and copy number variation were extracted. This genetic data molecularly characterizes the tumor at multiple levels, in this case 3 levels, as I mentioned. And then these tumor samples are then grafted into immunosuppressed mouse. And these immunosuppressed mouse and the tumor samples are administered several cancer drugs. Then the tumor's growth response to their respective drugs are documented. This growth response data and statistics records whether the tumor is sensitive or resistive to different treatments. Both the molecular characterization data and the treatment response data are made available along with the publication. And such a wealthy of data usually generates a lot of research questions. And my interest was specifically to use this data to design and implement machine learning models to predict tumors' response to different treatments. So out of the 5 cancers studied, I focused on breast cancer and colorectal cancer. And the gene expression data, the copy number variation data, and the gene mutation data for which there are total number of genes scanned, thousands and thousands of columns are the features from which the machine learning model defines a function to predict tumor's response to a drug. So the tumor's response here could be sensitive or resistive to a particular drug. So technically, for this particular study, I used random forest model as a binary classifier to predict the responses of the test set into sensitive or resistive. So what do we mean by that? So the training set of breast cancer and colorectal cancer involves multiple features, as I mentioned, and these features could be from gene mutation or gene copy number variation or genetic expression data, gene expression data. And so these features and these and the PDXs or the samples studied here are then classified into whether the samples are resistive or sensitive to the treatment administered. So we split this data into a training set and then let the machine learning model learn the underlying pattern within this feature set and their response and their relation to the response variable here, which is the treatment response, and then apply that machine learning model to a test set which was set aside and not to overfit the data. And then the predictions from the test set are reported. This is how usually a machine learning model is being built for the data set. So in this manner, the treatment responses of breast cancer and colorectal cancer are predicted for 13 separate treatment combinations for each one of them. So how do we then measure, or how do we then or quantify the performance of the machine learning model? So the machine learning model's performance can be quantified by identifying 4 measures, that is true positive, false positive, true negative, and false negative. So often precision and recall are used as accuracy measures. Precision can be defined as how many of the total predictions that we made were actually correct. And recall can be defined as how many of the total correct items were actually predicted correctly. So that is what is given in this very graphical schematic representation. I love this schematic representation. But for this study's purpose, I use a parameter called Matthews correlation coefficient, in short MCC, to study or to measure the accuracy of the prediction model or machine learning model. So why do I use this? So this metric actually uses all 4 performance metric. If you've seen this measure actually uses all 4 performance metric. If you see the formula, it uses true positive, true negative, false positive, and false negatives, all of them in the numerator. Mathematically, that is very important. And all of them in the denominator also. That gives this particular accuracy measure very nice mathematical properties. One of such properties is it is analogous to any correlation coefficient. So they lie between -1 to +1. So +1 would mean the machine learning model's predictor performance is perfect, and -1 would mean the machine learning model's predictor performance is very poor, and 0 would naturally mean it is random. There is no correlation at all. So it is random. So finally, how accurate was the machine learning model? So the performance of the model was compared with the traditional methods of predicting treatment response. So traditionally, single genes were used as biomarker that predict the onset of cancer. For example, you would know the BRCA genes mutations and other mutation data for breast cancer and other cancers were used to predict whether the patient is susceptible, or whether the person is susceptible to acquiring breast cancer or not. But these are biomarkers for the onset of the cancer, but they need not be the biomarkers or the signature genes for predicting the treatment response of that cancer. That is the problem that we have. So for many cancers, the same biomarker was employed to predict the treatment response. Naturally, this is not logical, and hence this is not an accurate predictor. So that is reflected in the performance in the left part of this graph, where the MCC curve for different multitude of treatment goes haywire, and none of them crosses even 0.5. And even if it crosses 0.5, they are very well known single gene pathway influencer for that particular cancer. And when I use, instead of one gene model, when I use all the genes to train the random forest model and to test, and I see a remarkable increase in the accuracy of the prediction. Yeah. So thus, we see that the treatment response could be predicted more accurately when we use data from multiple genes compared to traditional single gene markers. So, okay, we have a machine learning model that accurately classifies tumor responses, but we as scientists are not satisfied. We would like to know more, like we would like to know which set of genes were major contributors. So there comes a slight a dilemma. So as the quote sufficiently explains, there is a natural trade-off between the interpretability of the machine learning algorithm and its accuracy and in application. So in most literature on random forest model would tell you that the level of interpretation from genomic data is nearly impossible, since random forests are typically treated as a black box model. You put in data and you get out predictions and you forget about how that's been predicted and what is being used. So how do we implement the interpretability in this case, which is one of the key words in my title of the talk? So is there a better way to achieve the same performance with a limited number of genes, thereby increasing our chance of understanding the underlying biological mechanisms? Say, for example, I just plotted top 30 feature importances in this case, but actually it requires thousands and thousands of genes to match the predictions of the machine learning algorithm that was trained using all the genes. So one cannot naturally analyze thousands and thousands of genes for its pathway relationship with the cancer data. So what do we do now? So in general, there are only one way that we can go ahead in this case. So we have to use feature selection in this case. So feature selection by meaning we have to reduce the number of features or the number of genes in this case that the machine learning model learns the information or defines the function. So there are multitude of advantages of feature selection, and mainly they reduce the number of features. So one can, excuse me. So by reducing the number of features, one can reduce overfitting and also improve the generalization of the model. And then two, to gain a better understanding of the features and their relationship to the response variable. And how do we do that? In this data, this is the most complex step. So I employed a combination of clustering and univariate feature selection techniques to improve the interpretability of the machine learning model. First, the thousands and thousands, in this case 27,000 and something genes, are subjected to hierarchical clustering. So this removes redundant information by clustering similarly expressed genes together. The centroid of these similarly expressed gene pockets are then extracted, and then they are applied to univariate feature selection, which goes by ranking these centroids' relevance to the response variable, that is, to the sample's resistiveness or the sensitiveness. So by ranking them, we can use top 40 or top 50, in this case, a number that is empirically defined in this project. So the top 40 ranked genes for this model were selected, and then they were systematically analyzed for better performance, like iteratively, you go from top 2 ranked to top 3 ranked to top 4 ranked, and then that's to top 40 ranked genes to compare their performance to the performance of the machine learning model that uses all the genomic information data. So we do this. This is a complex system of reducing feature selection. So we do this because of two reasons. One, our input data as such has a lot of features, a lot of genes, like 27,000 genes. So traditional method of a greedy algorithm, which uses n into n, not n into n actually, n permutation or combination level of feature selection will not work here. It will take forever and ever to calculate that. And even methods like recursive feature selection and recursive feature elimination will not work here. That also will take a lot of time. So hence, such a method of combining hierarchical clustering and universal and univariate feature selection were employed in this case. So in the end of all this, we were left with around 30 to 40 most relevant genes instead of thousands. So with all this complexity, did I achieve the holy grail of balancing interpretability versus the machine learning model performance? So you be the judge. So you already saw the first two part of this graph, portions of this graph before. So when I include or when I employ this complex mode of feature selection algorithm, the accuracy improves for most of the cases. And in addition to that, not only that, in addition to that, when I employed or when I included information from the from other molecular features, like not only restricting myself to gene mutation, but also include copy number variation and gene expression data, the accuracy of the low scoring treatments could also be further strengthened when multiple multi-omics profiles like these are employed to determine a consensus of prediction. So all this means I could use all the data in my hand to accurately predict, and that accuracy is consistent also. As you can see, the improvement in the distribution of accuracy across the treatments from a data set that uses only single gene to a data set that uses all the multi-omics data, and in addition to that, in combination with the complex feature selection algorithm. So with this, now I have a handful of genes with their genomic data, and I can predict tumors' response to treatments with a reasonable certainty. And this is where this project stands as of now. So this handful of genes can further be used to identify their contribution in the tumor's response or tumor's resistance to different treatments. So in this case of 13 different treatments studied, some of the important conclusions that we obtained was there is no one size fit all method in cancer and the tumor and the treatment response prediction. So you have to use a specific machine learning model or specific model that is trained on specific data for specific combination. For example, you can see from this BGJ treatment, which is a very famous treatment for breast cancer, a single gene marker was sufficiently important right throughout the case, and it ran very well in these first two cases. It ran very well in all the compared to all the complexity of feature selection and increase in the information content by increasing the genomic data available. But for the other cases, I can push the prediction accuracy from absolutely negative to random to all of them are almost crossing 0.5, which in this case of limited data set is very, very high, very good. So in summary, I would like to tell you that I have shown how to apply machine learning models to multi-omics data to accurately predict tumor's response to any given treatment. So in this study, I found out that multi-gene predictors or machine learning models that employ multiple genomic information of drug response are more accurate compared to traditional single gene markers. So I also demonstrated how to improve the interpretability of the traditional machine learning models to answer more biologically relevant questions. So what more could be done in this case? So algorithmically, the level of multi-omics integration in this algorithm is very, very basic. Like you predict the treatment's response for the machine learning model that is being trained trained on different omics data, and in the end, you just build a consensus of the predictions from these three different models. So that is the most basic multi-omics integration at best. So one can explore various ways to integrate the multi-omics data even before subjecting it to the machine learning model algorithm, and then explore the possibility of building models on this integrated multi-omics data without sacrificing performance or interpretability. Scientifically, the limited set of high-ranking genes could further be subjected to pathway analysis or a similar analysis to understand the mechanisms of drug resistance on those treatment tumor pairs where the response is consistently resistant right throughout the different data sets that we have tried to model the machine learning or try to use to model the machine learning algorithm. So that concludes my talk and my share today. Thank you all for listening patiently, and I will be very happy to answer questions in this topic or in any other topic that interests you. Thank you very much. Over to you, Suprajit.
Thank you so much, Dr. Raghavendra. That was an amazing presentation, and I was thoroughly surprised and also mesmerized to learn that how machine learning algorithms can be implemented to analyze and to get certain signatures that we can actually correlate with what's going on inside the cell and draw conclusions, and ultimately also develop various treatment strategies. So that's a really amazing domain which we are entering, and where I'm sure that many students want to contribute as well. So we would like to go through the questions that we have in the chat section for you. So we have one question from Sharmeen, and she asks that what are the possible options to predict one biomarker for potential therapeutic target for cancer? For example, can we target only CD44V as the breast cancer stem cell for potential therapeutic target?
I mean, there are two possible plays here, Shamim. So therapeutic targets are they are already done. There is at least I think 2 drugs that are under trials that uses the therapeutic targets methods instead of the traditional all cytotoxic methods. But here I'm involved in actually predicting whether the cancer at what stage will respond positively to your therapeutic targets or not. So right now, in the current field, people have actually gone beyond trying to understand cancer on the basis of single gene biomarkers. So now people understand, and if you are interested, you can follow the research of Professor Nitin Baliga. He's amazing in this aspect. So he and others have actually demonstrated that not one gene, but a link of genes actually alter significant pathways in the tumor cells, and then they are responsible for converting these normal cells into abnormal or tumor cells. So in this case, all these genes that are responsible for this conversion must be targeted, at least in expression wise. That's what been shown in expression wise, or in any other genomic level, so that this conversion can be reversed, or at best this conversion can be changed into a stable disease with which we can manage. So that is the best that we can get so far. So people have actually gone beyond trying to identify single biomarker or one gene that is responsible for the onset of cancer. So I hope that answers your question. So maybe there is still, as I mentioned, there is still some classic cancer like breast cancers at ER positive. I think that is that gets onsetted by 1 or 2 mutations, or in case 3. But that is it. Multiple cancers are now have actually people have actually demonstrated that it's gone beyond that one gene biomarker level.
All right, thank you so much, Dr. Raghavendra.
I can see another one. Yes,
Yeah,
please go ahead.
We also have one more question in the chat section from one of our participants. This is from Shinjani. So she asks, can we use this multi-gene detection principle for simultaneously detecting the response of cancer cells as well as associated non-tumor cancer associated cells like CAFs, etc.?
I mean, this principle, one of the main issues I see as a data scientist today, not only in biological sciences, also in data science being applied to all the other fields as well, is the models that we built are tuned or actually hyper-tuned to the input data set and the test data set that has been input data set that's been supplemented and the test data set that has been validated. Once you go beyond the boundary, the scope of the model actually reduces. So one natural progression or procession from the study that I did is to collect all the breast cancer or colorectal cancer patients' data from TCGA and then try to apply our model on that. But there lies a problem. Their response might not be similar to our response, in spite of establishing that such a model system actually correlates very well with the TCGA patients. So this proposal that I'm actually making is currently under trial and currently under progress. Maybe I'm also interested to see what my peers in JNU are trying to find out, or will eventually find out, if one applies such a machine learning model to a completely different data set and what to find out. So the moment that you go beyond cancer cells, that is even increasing or more increasing the potential or scope of such a prediction. So if you do not supplement a machine learning model with the necessary input information, it will not learn the underlying patterns. So if you supplement, say, these are the cancer cells that we got, these are the non-cancer cells that we got, and these are the responses for these cells for the particular treatment. Now you tell me whether this treatment is more cytotoxic to the cancer cells or it is killing more non-cancer cells. So thereby I can make effective treatment predictions. So this could be a good starting point for a question like what Shinjini asks. But without supplementing the data of non-cancer cells or cancer-associated cells to the machine learning model, you cannot expect the model to predict response of those cells. I hope that answers your questions. If